every little foramen in the bone, every little process, etc. But it's pretty much rote memorization. Physiology is all function, because that's what physiology is, the study of function. So it's understanding processes. Micro is kind of a combination of the two. So we've started out with a lot of terminology and a lot of things. Cell parts, membrane parts. We have talked about some membrane functions. This section we're getting into now is very much about function. We're going to be getting into metabolic processes, not quite tonight, but we will be getting there. So as we go along, if you have questions, please ask them. And if you get completely lost, you can send me emails, you can ask me questions. I'll try to respond in a timely manner. Okay? So we're talking about microbial nutrition and growth. As I said, part of this is review, because we've covered some of this material when talking about criteria that are used for classification. So living organisms can be divided into 4 nutritional categories based on their sources of energy and carbon. You can think of this as kind of a review for the exam, because you need to know these 4 nutritional categories. So the first category, chemoheterotrophs. If organisms are chemoheterotrophs, they're using chemicals as their energy source, and they're using preformed organic compounds as their source of carbon. These organisms are sometimes referred to as chemoorganotrophs, which tells you, yes, they're using chemicals, and specifically they're using organic chemicals. We are chemoorganotrophs. We get our energy and carbon from the organic compounds we consume, which is rather significant, and it's something that you need to keep in mind, because we can't fix carbon. We're not capable of it. We're completely dependent upon another group of organisms that can build organic compounds using inorganic carbon. We can't do it. We have to eat. Then we have organisms that are chemoautotrophs. These are organisms that, again, are using chemicals for energy, but they can use inorganic compounds for carbon. In fact, very often, the chemoautotrophs use strictly inorganic chemicals for everything. Then they might be categorized as chemolithotrophs. Litho has to do with earth or rock, and it means that these things are using nutrient sources that probably most of us would never consider edible. The organisms that can do this, as far as I know, are all prokaryotic, and some of them live in some really strange habitats. You can see a hot spring here. You can see some thermal vents on the seafloor. This is a rather unusual nutritional category. Nevertheless, it exists. Then there are organisms that are photoheterotrophs. If photo is the first part of the word, that means they're using light as their energy source. But then it's followed by hetero, which has to do with their carbon source. The photoheterotrophs use light energy, but they can still use organic compounds for carbon. Again, as far as I know, the only organisms that fall into this category are prokaryotic. And then the last category, photoautotrophs. These are organisms that are using light as their energy source, and they're using inorganic compounds for carbon. Notice that I made photoautotroph green. Why?
Green algae.
Well, so green algae fall into this category, cyanobacteria fall into this category, plants fall into this category. By and large, organisms that use light as their energy source have to have some kind of light trapping pigments, and a lot of times those pigments are either chlorophylls, which are green, or bacteria chlorophylls, which are sometimes green. Of course, then there's bacteria rhodopsin that's not green at all, it's pink, but anyway, that's why photoautotrophs was green, yes. Well, so for this laboratory activity, in fact, for our working with bacteria in this laboratory, we're primarily interested in the chemoheterotrophs. These are the organisms that are most likely to be in the clinical setting. They're the most likely to be encountered in the laboratory. If you take a plate home and expose it to the air, they're the ones that are going to show up growing on that plate. It turns out that not all of these can be grown in vitro, but for the ones that we work with, the nutritional category they're most likely to be in is chemoheterotroph. As I say that, and I think, well, we do have one morphological unknown, the little rhodospirillum. The rhodospirillum is actually a photoheterotroph, so they can use light as an energy source. On the other hand, if you grow them in a dark cupboard, then they're not using light. They're capable of using light, but they don't have to. Now, another thing that I mentioned earlier is that when we try to grow bacteria in vitro, in an artificial container, it is really very artificial, because bacteria typically grow in communities where they're interacting with other organisms. A lot of them are symbiotic. They live inside plants or animals or some other kind of organism. Consequently, the majority of bacteria, and this is something that people are just determining, the majority of them cannot be grown in vitro. Well, maybe they're dependent upon some other organism type to produce an enzyme that they need to break down a food source. Or maybe somebody else in the community is actually producing a substance they're using for food. So a lot of them can't grow by themselves. Consequently, we're looking at a very limited group of organisms, not just chemoheterotrophs, but only a few of those. Still, as I said, these are most likely going to be the ones of clinical significance. They're the most likely ones because they can live on or inside our bodies. They're the ones that are most likely to be etiological agents. So, they are of considerable interest to microbiologists. Well, okay, so as I go through and talk about growth factors and so forth, mostly we're talking about chemoheterotrophs, and specifically chemoheterotrophs that will grow in vitro. It used to be people thought we knew so much about bacteria, and it's only recently when people have done what are called next generation sequencing, where they're sequencing from communities, that they are really getting the information that says, you know what? We know almost nothing. What we know is based on just this small group of organisms, and there are so many other bacteria that cannot be grown this way. So the other thing is we're going to be talking about microbial growth. So when we talk about growth as it applies to microorganisms in general, we're referring to an increase in cell number rather than to an increase in cell size or organism size. That's not too surprising, because if you think about growth of a multicellular organism such as your cell, did you not start as one cell? And so growth involved an increase in cell number. An organism gets bigger by increasing in cell number. You compare that to bacteria. One cell lands on the surface of an agar plate. That cell begins to take in nutrient, and it begins to reproduce, and you get more cells. You cannot see cells on an agar surface, but what can you see? Colonies. And a colony is a mass formed by a cell reproducing itself many, many times over. So it's not that different. So any single bacterium that is placed on the surface, and again, pay attention to the endings on words. Bacterium is singular. If you use the word bacteria, it is plural. You can't have a bacteria. Neither can you have a fungi. Fungi is plural. Bacteria is plural. Anyway, a cell that lands on the surface, let's say we have a nutrient agar plate, you put a cell on there, you're not going to be able to see that cell, but it's going to be taking in nutrient, and pretty soon it'll grow to form a colony. Consequently, one cell is equivalent to what we call a colony forming unit, which I didn't write. Instead, I used the little abbreviation CFU. CFU stands for colony forming unit. As soon as I say that, and I think, sometimes there can be exceptions to this because, well, for example, if you take a look at this illustration, you see these cells mostly arranged in tetrads, 4 cells, and they tend to be fairly stable. Consequently, if you were growing this organism type, probably a single colony forming unit wouldn't be 1 cell, it would be 4 cells in a tetrad. So there could be variation. But a lot of bacteria that we work with in this laboratory have single cell arrangement, particularly if you put them in broth, and oftentimes we're transferring cultures from broth. In broth, the cells are separated, so each cell in contact with the surface is a colony forming unit, or CFU. There we see a nutrient agar plate, and then you see the nutrient agar plate after it's been exposed to air. So you see lots of different colonies. Again, probably each one of those colonies was initiated by one cell making contact with the agar surface and then reproducing itself. If you are growing bacteria in broth, there are other things to look for to indicate that you're going to get growth. You're not going to be seeing colonies in broth media. as soon as I say that, I'm thinking, well, you can. If you have streptomyces, the filamentous type bacteria, they form these masses of threads, very much like hyphae. If you put them in broth and put them on a shaker, the shaker is constantly rolling those thread balls, and they will form little balls, essentially little round colonies in there. They're just masses of hyphae all tangled up. But usually we can see growth in a broth medium as turbidity or cloudiness. Sometimes that turbidity is fairly uniform. Sometimes there will be more growth toward the top or toward the bottom. If you have a layer on the top, this is called a pellicle. Sometimes a pellicle is a skin-like layer. So if you get a culture and you see a layer of cells right on the top, that layer is called a pellicle. And then the other thing that can form is called a sediment. A lot of times bacteria that are not motile will form a sediment. They'll all sink to the bottom. Then again, if you leave them sitting in the refrigerator for a long time, if you remember when we took our morphological unknowns out initially, they were almost all sitting on the bottom. But they had been put in there, and then they sat in those tubes in the medium for about 2 weeks before you guys got them. So they all sat on the bottom. The other thing to remember is that growth in living organisms, growth involves assimilation of materials from the environment, and assimilation requires metabolism. So metabolism, all the chemical reactions, that means food materials are going to be taken apart, that's catabolism, and then those small pieces that are formed are going to be used to build new cell parts, so that's anabolism. So growth is tied to metabolism, which is tied to assimilation. These are just pictures of bacteria.
Like right under before, I know.
So again, the nutritional categories, the 4 nutritional categories, as far as I know, prokaryotic cells are the only organisms that fit into all 4 categories. That doesn't mean the same type of prokaryotic cell fits into all 4 categories, but prokaryotes in general, bacteria and archaea, fill up all those categories. Still, we're going to concentrate on chemoheterotrophs. These are the organisms that we can grow in the laboratory in vitro. And in vitro means what again? In an artificial container. So an artificial container, it can be a plastic petri plate, it can be a glass tube, it can be a flask, it could be a fermentation chamber that might be as large as this room. Sometimes bacteria are grown under conditions where they have really huge vats of media. Well, so that then brings us to what is the culture medium. Remember, culture medium is a mixture of materials that provide all the nutrients organisms need to grow in vitro. And we looked at some features of culture media earlier. This was in laboratory. We talked about defined media, where the exact constituents are known. You have to put in specified quantities of very specific ingredients to make defined media. And then there were complex media, where you don't really know what's in it. Anytime you see a medium container that says it contains extract, it can be beef extract, yeast extract, soy extract. If it's an extract, you don't really know what's there. You know you've got some protein, some lipids, some carbohydrate, but you don't know the exact constituents or the amounts. The other thing we looked at was solid versus broth media. What makes media solid? Agar, the solidifying agent that we use. We don't normally use gelatin or starch as a solidifying agent, and the primary reason we don't is because some of the bacteria we work with in the laboratory will digest those things. So if you had a plate that was solid, but it was made with gelatin and the bacteria ate it, it would just be soup. The same thing can happen with starch. Even, we do have some media that we'll use later in the semester called starch agar. It has starch in it, but the solidifying agent is still agar. The other thing about gelatin, of course, is it becomes liquid when you warm it up, which agar won't. Well, it will if you get it hot enough. But you have to get it up near boiling, and then it will. So what we said about media is that media typically have to contain certain constituents. So usually there's a carbon source, usually there's a nitrogen source. Of course, right away you can say, what organisms don't need a carbon source? Anything that's an autotroph. An autotroph can get carbon from the air, carbon dioxide. You don't have to give them carbon. And who can get along without a nitrogen source? Anything that can fix nitrogen. So we talked about Azotobacter with our soil enrichment, but Rhizobium, Agrobacterium, actually there are lots of bacteria that can fix nitrogen. That means they can take N2 molecular nitrogen from the air and use that. Initially they make nitrate, nitrite, ammonia, but then they can use that nitrogen in the form of inorganic compounds to make proteins, nucleic acids, etc. And then typically you have to have some minerals. Sulfur, as I mentioned, sulfur is often used to tie together polypeptide chains in quaternary proteins. A lot of times there will be a sulfur associated with one or another amino acid, and then they can hold the chains together. Another mineral is phosphorus. Where did we see phosphorus used? In cell membrane, in specifically what kind of molecules? Phospholipids. Remember, you've got two fatty acid tails and a phosphate group attached to a glycerol backbone. So phosphorus is used in cell membranes. Where else is it used? Remember nucleotides? Sugar, nitrogenous base, phosphate. So you have to have phosphorus in your nucleotides to make nucleic acids as well. And then there are minerals such as iron and calcium and magnesium. These are often cofactors. Cofactors are enzyme helpers. We'll talk more about these when we get into metabolism. Exactly what do we mean by an enzyme helper? Enzymes are proteins. The helpers are not. So cofactors are not proteins. They're very often minerals. And there's a lot more of them than these. So manganese, copper. And copper is not one of the constituents when we talk about our C. Hopkins cafe, mighty good if you have to add salt. Copper isn't listed, but copper can still be an important cofactor. And even molybdenum can be. And of course you have to have water. Water is going to influence tonicity. We talked about the amount of solute that's in an environment. You have to have water. Water is necessary for metabolic activity because water is involved in hydrolysis reactions and condensation reactions. If you have very high amounts of water, which is low solute, if you have very low solute in your environment, what do we call that environment? In terms of tonicity, hypotonic. So if you have low solute, it's hypotonic. And if a cell is placed in a hypotonic environment, water's going to come in. Depending on what kind of cell it is, it might just blow up. Red blood cells will. Bacteria won't because they have walls. Algae won't. Fungi won't. If you put them in a hypertonic, hyper means high solute, then the cells are going to tend to lose water. You need to know those terms and know which direction the water is going to move. If it's isotonic, you have the same amount of solute inside the cell as relative to the environment. The other thing I talked about relative to media was that some of our media contain buffers. Buffers are just substances that resist pH change. We put buffers in if we don't want the pH to change because it'll slow down metabolic activity. It might even stop growth. And if we don't put in buffers, oftentimes we'll put in pH indicators. That's what you see in the images here. pH indicators make the media different colors. So we will be using phenol red, bromethymol blue, bromcresol purple, methyl red, neutral red in one case, but not usually. Anyway, pH indicators, they make our enzymatic test more interesting. When we start physiological unknown 1, you're going to be using a lot of different media that come in various colors because we put pH indicators into them. Now, all of that was reviewed. Hopefully it sounded familiar to you.
Yes.
Hopefully you could say, oh yeah, I've been over that stuff. So those terms sound familiar. This term, probably not. Microorganisms are referred to as fastidious if they require some specialized growth factor. Sometimes we're not even sure exactly what it is that the microorganisms require, because oftentimes if they're fastidious, they're going to require some kind of vitamin or some kind of mineral that is in a constituent of tissue, and we can't necessarily define it. Sometimes the fastidious microbes are referred to as picky eaters. So they need something, and if you don't give it to them, they simply will not grow. We grow fastidious microbes in this lab by using media that is called enriched. Be careful of the terminology here, because enriched medium is not the same as enrichment medium. An enrichment medium is you're trying to grow one kind of thing while you're inhibiting the growth of something else. Enriched media are those that contain cells or tissue fragments. This plate that you see here that has a very bright red color, this is blood agar. We have blood agar plates in here, and they're made with 5% sheep blood. Blood contains nutrients. We don't necessarily know exactly what's in there, but if you don't have whole red blood cells, certain organisms will not grow. And those are the fastidious microbes. Some of the ones that we grow on blood agar in here are Streptococcus. Streptococcus pyogenes, Streptococcus orales. There are quite a number of Streptococcus organisms. If you put them on nutrient agar, they won't do anything. I mean, you can streak a plate, but you're not going to see any growth at all because they're fastidious and they require this enriched medium. Some of them can grow on brain heart agar. Brain heart has a little brain, a little heart. Again, tissue fragments. You don't know exactly what's in there. That's a complex medium. Enriched media are complex. You don't know exactly what's there. Neisseria is another fastidious microbe. They will grow on brain heart agar. Enterococcus will grow on brain heart agar. Raffia will grow on brain heart agar. But if you put them on nutrient or TSA, they probably won't do anything. Well, so then we can look at the various factors that influence microbial growth. And again, this is material that we've gone over because we put this under the heading of factors that can be used for classifying. So here I'm going to be asking you some questions. This is actually an exam review. So, gas requirements. If we look at gas requirements, we know that we have a whole bunch of categories based on gas requirements. What are the categories? If they have to have oxygen, we call them obligately aerobic or obligate aerobes. If they can't tolerate oxygen, we call them obligate anaerobes, or we say they are obligately anaerobic. And if they can live either with or without oxygen, we call them facultative. It used to be always facultative anaerobes. Now you'll see references calling them facultative anaerobes or facultative aerobes. If they're facultative, they can go either way. The other category was microaerophils. We don't have any of those for the laboratory, so I tend to ignore them. So then, temperature requirements. Again, categories and a bunch of terms. What were our temperature requirement categories? Starting at the cold end. Psychrophiles, so -5 to 23 centigrade psychrophiles. They grow best in cold. And then if you warm it up and you get between 20 and 45 degrees, we call them mesophiles. Mesophiles. So if I say between 20 and 45, right in the middle of that is our incubator temperature, which is 37. So the organisms, most of the organisms we grow in here, we either grow in the incubator at 37 degrees centigrade, or we grow them in the room temperature, which is somewhere around 25, 26 degrees centigrade. Mesophiles. Yes. If they grow in you, yes, they're mesophiles. Consequently, mesophiles are the most likely to be etiological agents, the ones that are most likely to infect you. Mesophiles. Bring the temperature up some more, 45 to 60. Then we have thermophiles. The name means warm loving, but if you've ever put your hand in 60 degrees centigrade water, You're not gonna leave it there for any length of time because it is really hot. It's just that some microbes can grow even in water that's hotter than that. You get up around 90 degrees, then you have, what kind? Hyperthermophiles. Something like Thermus aquaticus, the ones that live in the hot springs. They really like it hot. The Thermus aquaticus produces an enzyme that is used to replicate DNA, and that enzyme works best at 72 degrees centigrade. That's really hot, but that's the temperature they like best. So hyperthermophiles really like it hot. What do we call them if they can tolerate exposure to cold? They don't die, they don't grow either because their metabolism is stopped, but if they can tolerate exposure to cold, they are cycloduric. Most bacteria are cycloduric. And if they can tolerate exposure to heat without dying, thermoduric. Endospores are thermoduric. Okay, again, this is kind of a review. If it doesn't feel like review, you need to study more. These are all terms that you should have been learning as we were going along. They're kind of the basis. We keep sticking them in here as applied to different groups of organisms. pH requirements, the only terms that I talked about relative to pH, I said if organisms really like an acidic pH, we call them acidophiles. There is the category of basophiles, but what I said about that is it's spelled the same way basophil is spelled, and those are white blood cells, so never mind. pH is a measure of acidity versus alkalinity. If the organisms like a neutral pH, that's around 7, usually between 6.8 and 7.2. Most of our media start out at that pH. And to keep the pH constant, we put in buffers, and if we want to see the pH change, we put in pH indicators. Osmotic pressure requirements, we just went through this. Hypertonic, hypotonic, isotonic. Again, you should be familiar with those terms. Symbiosis. This is an important term. You need to know what is symbiosis. When you have organisms, 2 or more different types of organisms living together in close association. It's kind of funny to me to think about the fact that biologists used to think symbiosis was rare. And they'd get all excited if they could find one example of symbiosis, or maybe 2 or 3 examples. They just weren't looking, because we are symbionts. We have a multitude of organisms that live with us, inside us, outside us, on our surfaces, that we cannot survive without. So we are dependent upon our symbionts in many, many ways, and probably most ways we don't even know, because we don't know what they are, who they are. Anytime somebody tells you you should be killing 99.9% of them, just go, I don't think so. And then I think of those Star Trek episodes where they have those devices that take the person from one spot to another spot by scrambling their molecules, and anyway. If they didn't bring your microbes with you when you did that, when you got to the other end, you just died. It'd be sad. Anyway, so there are lots of different symbiotic categories. So you can have mutualistic, actually most symbiotic relationships are mutualistic, everybody benefits. But parasites are symbiotic. They're dependent upon a host, they're taking from the host, they're not giving anything back. Pathogens, that's another symbiotic relationship where the pathogen is dependent upon the host. For a while, a lot of pathogens kill off their hosts. You think, well, if it was really the best symbiotic relationship, eventually pathogen and host would reach a balance, and they would stop killing their hosts. It's not in their best interest to kill off their food supply. Anyway, and then I mentioned, if organisms typically grow in symbiosis, that is, they're dependent upon some other organism that's living with them, when you take them and put them by themselves, they don't do well. It's not surprising. They can't grow. Well, okay. Now we're going to talk about how bacteria reproduce. This is going to be reminiscent of talking about how fungi reproduce and how algae reproduce. But it's important because we know that abiogenesis isn't valid. Abiogenesis is what? Spontaneous generation, which means it means living organisms can arise from non-living material. As far as we know, the cells that we encounter now are not arising from non-living material. They're coming from other cells. The process is biogenesis. And that's what we're going to talk about. How do bacteria reproduce themselves? They use biogenesis. One cell gives rise to daughters. They undergo a process called binary fission. I mentioned binary fission versus longitudinal fission. If I have a cell that is a rod shape, what's binary fission? Across the long axis. So binary, you're cutting the cell in half. Pretty much bacteria tend to reproduce by binary fission. There are some exceptions, but most of them do. And even cocci will elongate a little bit and then divide across the long axis. Eukaryotic cells undergo meiosis, they undergo meiosis, yes, but they undergo mitosis also. If you take a general biology class, you spend quite a bit of time talking about mitosis, and I never do in this class, and the reason is because bacteria don't do it. Mitosis is the separation of the chromosomes, the business where the chromosomes line up down the center of the cell. The spindle apparatus is formed, which is microtubules, and then the chromosomes are pulled apart. Bacteria don't do that. There's a whole bunch of different phases of mitosis. Never mind, bacteria don't do that. So here we see some bacteria undergoing fission. This image, I don't know if you could find the film, but you might look, if you Google bacterial fission and look for videos or look for films, you might actually be able to find films that shows how the cells come apart. This is called a snapping arrangement, where the cells are sitting like this, and they actually snap apart. So when they get ready to split, something happens in here where they, presumably what happens is the cell wall is, two pieces of the wall are flat, and then they become somewhat convex and push away from one another, and they actually pop apart. There's some really interesting images that people are beginning to get now. Some of it is computer-assisted microscopy, so that they can see things that they couldn't have seen before, sometimes because they can use fluorescent dyes and make things visible. But it's just really interesting to see some of the images that are now being prepared relative to bacteria. Well, okay, so as you can imagine, fission is going to involve a series of events, and in all biology classes, we tend to divide events up into fragments, and they'll put names on each fragment. So we do that all the time. Take a process, chop it into a bunch of little pieces, and then put names on the faces. So the first thing that has to happen, if bacteria are getting ready to undergo fission, you know that one cell is going to become two cells, and what is most important to maintain is the genetic composition. You have to have the genetic information that is going to maintain cell function. So before a cell can begin to undergo fission, it has to replicate its DNA. This process is kind of interesting, and we'll talk in a great deal of detail about DNA replication, probably more than you want, and yet we're just scratching the surface. Have you ever skipped a rock across water? Did I mention to you that that's the amount of information that you get in this class? Imagine the lake, a very deep lake. You're just touching the surface. Everything we do in here is just touching the surface. Well, anyway, so E. coli. E. coli has a chromosome that, if you stretched it out, is about 1 millimeter in length. Well, remember, E. coli, a single E. coli cell is about 1 micron in diameter. They might be 2 to 3 microns in length, but about 1 micron in diameter. I'm holding it up for you. But anyway, the chromosome is about 1 millimeter, so it's significantly bigger than the size of the cell, but it's just very, very long. It takes about 60 minutes for an E. coli cell to replicate its DNA. As I said, we're gonna get into the section on genetics where we talk about how replication occurs. At this point, just recognize replication means copying the DNA, making new DNA. Well, an interesting feature about E. coli, when E. coli cells are growing under optimal conditions, they can undergo fission every 20 minutes. Now, if you look at that, that presents a bit of a problem. The chromosome must be replicated. You must duplicate the chromosome in order for each daughter cell to get a complete copy. If it takes 60 minutes to replicate the entire chromosome, how can they undergo fission every 20 minutes? Any guesses? And have you heard of this situation before? Maybe? Kind of creates a conundrum. Well, so this is the chromosome made very simple so that you see it is a covalently closed circle. CCC DNA. Replication starts at one point. It's called the origin of replication, and it proceeds in both directions around this loop. Here you can see replication starting, and the red indicates the new DNA that is being built. The black is the old DNA. So it started at a point and it's replicating around the loop. What they do is begin a second replication cycle and a third replication cycle before the first one is complete. So they start one, it begins to go. Before that gets to the bottom, they start another one. Before that gets to the bottom, they start another one. So they're running 3 replication cycles at the same time. And this is how they can undergo fission every 20 minutes and everybody gets a complete copy of the chromosome. Which means they're running DNA replication pretty much continuously. When we get to the section on DNA and what DNA controls in the cell, which is basically all of metabolism, by telling the cell what enzymes it can make, you gotta somehow coordinate copying sections of DNA to make RNA that will encode enzymes. At the same time, replication is happening. It's kind of crazy. Anyway, so again, before one replication cycle is completed, a second cycle, and then a third cycle will start. Basically, you take 60 minutes and divide it by 3. And that's how they can undergo fission every 20 minutes. The next thing that the bacteria have to do is elongate. Even if they're coxy, as I said, they will elongate just a little bit. But this also creates a kind of a puzzle because they have to elongate, and yet peptidoglycan is not elastic. It does not stretch. It is a solid molecule. We talked about the composition of peptidoglycan, N-acetylglucosamine, and acetylmuramic acid in polysaccharide chains, tied together by little short peptides. If it's a gram-positive type cell, you have many, many layers of this stuff. It does not stretch. How do they elongate? Well, it turns out what they have to do is take apart that wall just a little bit in sections. If they are rod-shaped cells, typically what they will do is have multiple sections along the length of the cell. So if this is a rod-shaped cell, there will be multiple regions. Can you see that if it's orange? Mm-hmm. These would be the regions where elongation can occur. And you can visualize these regions as being kind of like the epiphyseal plates that allow long bones to increase in length, where basically what happens is the peptidoglycan is taken apart. And as soon as it's taken apart a little bit, the fact that water tends to come into the cell will cause it to stretch, and then they really quick rebuild the peptidoglycan so that they don't fall apart. If you had a brick wall and you wanted to elongate it, the equivalent would be to take a section of wall, take it apart, and then you got to slide both portions of the wall away from each other. It wouldn't work so well because walls are sitting on the ground. But anyway, yeah, they have to partially decompose the wall and deposit new peptidoglycan. If they are coxy, there's typically one region where they do this, right around the equator, and they only have that one space where they deposit new wall material. They have to take it apart, and then they have to build new wall. Yeah. like I said, for bricks, that would be harder. Now, there are some differences in bacterial diameter, but most of their growth is going to be in length. They tend to elongate. And again, this is taking the wall apart and depositing new walls. Why is this significant? Well, later on, we're going to be talking about how antimicrobial drugs work. And the drugs that are called beta-lactam antibiotics, which include penicillins and cephalosporins, those drugs prevent the formation of peptidoglycan. Why do they kill bacteria? Because bacteria take their wall apart to grow. And if they take their wall apart and they can't build no new wall, they die. So it does matter that we understand how bacteria function in order to understand how can you make them not function or make them die if that's what you want. So after the cell has elongated, the next thing that's going to happen is the membrane is going to fold inward and form a septum. Again, a septum is just a wall that's going to be across the long axis of the cell. It's going to separate the cytoplasm into two parts. Here you can see an electron photomicrograph showing a septum beginning to form. So the membrane is coming inward. It's going to come in like this. And ultimately, it's going to come all the way across. So you're going to have a double layer of membrane separating the two pieces of the cell. Once the septum has formed, then wall material is going to be deposited. These images, again, there are some interesting images that can be obtained now because we have new technology. These images were made using fluorescent dyes and fluorescent microscopy. And so there's a certain type of protein, different types of proteins, but the green one is called FITZ protein. Those determine where the septum is going to form. They're involved in septum formation. So they actually form these rings, and then the membrane squeezes together and forms the septum. Once the septum is complete, as I said, you have a double layer of membrane, then wall material gets deposited in the septum, outside the membrane, so it ends up being between the two membrane layers. So it's in the septum, between the two cell parts. And then the last thing that's going to happen is the cells will physically separate. Sometimes it's a popping action. Sometimes, well, if you watch, and we do have an animation in a little bit, if you're watching cells that are bacilli, sometimes they come apart, but rather than coming apart and moving away from one another, they'll come apart and then slide across like this. They tend to want to maintain contact. Then those will undergo fission and again slide across so that they're all in contact with one another. They tend to do that on solid media, which is why they'll form discrete colonies. The cells are all piled up together. There seems to be quite a bit of communication between cells during these processes, but I've also seen some really interesting footage showing bacterial colonies that as they begin to grow close to one another, the bacteria jump back and forth between them, which is like, what? They're doing some kind of communication between colonies too. So these steps, you want to be able to put these steps in order. A question that you might see on next week's quiz would be something along the lines of, what is the sequence of steps in the fission process? Here we're going to see an animation. You see one cell on the right, and then a cell that has undergone fission on the left. Watch these cells continue to divide.
By doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
Now, I'm going to show that again because I didn't have the sound going initially.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two.
But not that rapidly.
Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
So you can imagine this is forming a colony then, and you can see that mass of cells. You can find this kind of video showing bacteria, and typically there will be a soundtrack that says bacteria divide very rapidly. Not that rapidly. They're not that fast. But how rapidly can they divide? It turns out pretty fast. However, before we get to that part, because when you get into the growth curve business, when we are looking at bacteria, and some of you have finished, almost said a lot of you have finished, a few of you have finished your morphological unknown, so you had to talk about something having to do with what happens to bacteria if they don't physically separate. So remember, you did an indirect stain and you were supposed to record size, shape, and arrangement. So if the cells don't separate, and I tried to pick an image that showed some arrangements. So here we have cells in chains. Those are strepto, in that case streptococci. Cells in chains are strepto. Here we have two cells connected, that's diplobacilli. Those on the other side kind of look like staphylo. You get these arrangements when the cells don't physically separate. If they're in broth, very often they will, but some don't. Some tend to prefer to maintain connections, and then you will get specific arrangements. So we can look at how those arrangements come to be. Again, strepto, staphylo, those are arrangements. So if we had cells, whether they were cocci or bacilli, and if they were to undergo several separations, but the fission plane always stays the same, then what kind of an arrangement are you going to get? Strepto. So in this case, what is the orientation of the fission plane? It's always across the long axis, but it's not changing. So it's always this way. Fission plane is here on the top and down here. If the fission plane always stays the same, then as the cells continue to divide, you're going to get strepto, either streptobacilli or streptococci. Consequently, those terms are, well, streptobacilli is actually not a genus name, but streptococcus is. So now we're looking at one coccus-shaped cell, one spherical shape. Let's say that the fission plane is going to change, and we're going to allow this cell to undergo 3 division cycles. Then what will the arrangement be? We have one cell, the fission plane is vertical, then the fission chain changes to horizontal, and then the fission plane changes to this way. So it was this way, then 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 it was this way. But what is that arrangement on the end? No? That's very specific. It's a cubical packet. Anybody remember? Cubical packet is called sarcine. And we have some of our morphological unknowns that do this. So when you look at them, they are in clusters that appear to be cube-shaped. Definitely cubes, not random break-like clusters. I'll bet there'd be some commercial interest in growing grapes in sarcine, but as far as I never did one do that. They could package them up really easily. But bacteria do. So sarcine is these cubical packets, 8 cells. If the fission plane changes randomly, keeps changing every time they undergo fission, then you get staphylococcus. So 1 becomes 2, 2 becomes 4, and then 4 become this kind of random break-up cluster. That's staphylococcus. Well, okay, so if they don't completely separate, then depending on how the fission plane, either if it stays the same, you're going to get strepto. If it changes, you're going to get some other kind of arrangement. Now, we can look at what's called population dynamics in a batch culture. When we're dealing with bacteria and yeast also, where you're putting a population of cells into a container, they can be in a flask, they can be in a tube, but if you put them in, and that's the only nutrient they have available to them, then we say they are grown in a batch culture. We grow batch cultures a lot in here. Put the cells in a tube, let them grow. So they're in vitro, and they're in a container that's closed. Here we see different examples of batch cultures. Some are in tubes, some are in a flask. if you were to look at what's going on with that population over time, what you'll see is that they tend to undergo changes in density, the number of cells per volume of liquid in the batch culture, that is pretty much consistent with any given type of bacteria. Every time you put them into a batch culture, they will do the same kind of thing. And pretty much all populations tend to follow this pattern. So they will cause or produce a growth curve that is predictable. You can expect it to look a certain way. In this case, the vertical axis is a logarithmic scale. The horizontal axis is not. It's time in minutes or hours. And this is the series of phases. So this is called the lag phase, exponential growth phase, stationary phase, and death phase. So we look at these in detail and see what's going on with the population. And again, some variation is going to be expected. It depends, like if you put bacteria from, say you have them growing in tryptic soy broth, and then you put them in a new batch of tryptic soy broth, their lag phase is going to be really short because they're already geared up and ready to use that particular type of medium. Again, the vertical axis is logarithmic. So we're expressing population density as log, the logarithm of the number of cells per milliliter, or colony forming unit, CFU per milliliter. So our lag phase. Lag phase is a straight horizontal line. We look at it here. The length of lag phase depends on what the cells were growing in initially. As I said, if you take them out of tryptic soy broth and put them into a new batch of tryptic soy broth, they're going to have a very short lag phase. If you take them from some medium that is completely different and put them into tryptic soy broth, so in order to use it, they have to make a whole bunch of new enzymes, then their lag phase is going to be longer. But again, no increase in number during the lag phase. So what's happening? These cells are getting ready to grow. But growth, remember, means increase in number. So what we're looking at is all the things the cells were doing prior to physical separation. So there's going to be an increase in DNA content because the DNA has to be replicated. There's going to be an increase in metabolic activity because in order to reproduce their DNA, they have to metabolize. They're making a bunch of RNA. They're making a bunch of protein too. They're going to increase in size. They elongate. So they're going to increase in size, and they increase in dry weight because they got bigger. So all those things are going to be happening, but no increase in cell number during lag phase. We can think of the cells as gearing up for growth. They're getting ready to start the fission process, and once they begin, again, they can undergo fission fairly rapidly. So the next phase is called the exponential growth phase or logarithmic growth phase. If you're using a log scale, the exponential growth phase is a diagonal line. They're going to be increasing number fairly quickly, depending on the organism type. But during this logarithmic phase or exponential phase, typically they're reproducing very rapidly. The culture is metabolically very active. They're using up a lot of nutrient. So we say they're reproducing exponentially. One cell is going to become 2, and then 2 become 4, 4 become 8, 8 becomes 16, and then 32 and 64, and 128, 256, etc., etc. You can imagine that as they get more numerous, every time they undergo fission, they produce a tremendous number of cells because they're just dividing, each single cell producing 2. The time that's required for the cells to complete the fission process, that is to go from 1 cell to 2 cells, is called their generation time. Sometimes it's called doubling time. So generation time or doubling time. Generation, if you think you have a parent cell that divides in half and produces 2 daughter cells. But then if you think about bacteria, and that's how they reproduce, you don't want to think too long about how old are they. Because if you had a parent cell and it divides in half and you've got 2 daughter cells, those are the same age, and then the next generation. But how old are they? Very, very old. What's interesting is that generation time or doubling time can be 20 minutes if the cells are growing under optimal conditions. As I said, E. coli can undergo fission every 20 minutes. Staphylococcus can do this. Salmonella can do this. So it's really short. This is why if you have bacteria in the bloodstream, and if the immune system of the patient is not dealing with them appropriately, they can get to high enough numbers to kill the person very quickly because they reproduce so rapidly by exponential growth. It's been estimated that if bacteria could be maintained in the exponential growth phase for 45 hours, they'd form a mass the size of the Earth. Interesting idea. Can they? Really? No, they cannot. But if they could form a mass the size of the Earth in 45 hours, 20 minutes before that, the mass was half that size, half the size of the Earth. 20 minutes before that, it was only a quarter the size of the Earth. So it's when you get to those later phases where you have so many cells reproducing. Nevertheless, no, it's not possible. Why not?
The salt water. The salt water.
The salt water.
And the humans. And the drugs. And the soap. No.
Suppose there weren't any humans.
Or there wouldn't be at that point. Okay.
Probably not, no, because there'd be nothing left but bacteria. But what are they going to run out of?
Nutrients.
Nutrients. We're talking chemoheterotrophs. What are they going to eat? Well, first they eat all the humans. So no, they can't do that. They're not going to form a mass the size of the Earth. We'll come back to that in just a moment. So instead, they are only in exponential phase for a period of time, and then they go into stationary phase. And during the stationary phase, there's no overall increase or decrease in cell number. You get another straight line, a horizontal line. So this is stationary phase. The population growth is now stationary. Well, what does it really mean? During stationary phase, the number of cells dying is equal to the number of new cells being produced. If you have bacteria in a flask, you might think that that population is homogeneous, but I'm sure it is not relative to nutrient. There's going to be a little bit more nutrient in some areas than others. Some cells are still able to get enough nutrient to reproduce, while other cells are dying. So the number of dying is equal to the number being formed. The population has now reached what's called the maximum density or maximum concentration. Sometimes it's just abbreviated as M concentration. M concentration means maximum concentration. That's the maximum number of cells you're going to get. And it tends to be consistent for any population of organisms grown in vitro. And it is 10 to the 9th cells per mil. 10 to the 9th. Now, the reason they can't go above that number is because that batch culture has what we call a carrying capacity. But it turns out all environments have a carrying capacity. We can superimpose carrying capacity on this graph. It's the straight line coming across here, and K stands for carrying capacity. It turns out all environments have one. So carrying capacity, it's the number of organisms an environment can support, presumably in a fully functional state. It's not like how many people can you shove into a Volkswagen, or how many people can you squish into a phone booth. They can't function in there. We're talking about how many organisms can continue to function fully that the environment is supporting. So it's influenced by limiting factors. And limiting factors are factors essential for microbial growth, but present in limited supply. What immediately comes to my mind is nutrients. Why can't bacteria form a mass the size of the Earth? Because they're going to run out of food. The entire planet is not a food supply for them. Besides, part of it is molten. It's pretty hot. But things that would be limiting factors, they could be nutrient, they could be water. You can imagine water would be a limiting factor to the growth of trout. If you have a pond and rain stops and the pond dries up, trout are not going to do so well. And the same thing can happen to microorganisms. Temperature can be a limiting factor. Oxygen availability. lots of different things. So it isn't necessarily just nutrient. But every environment has a carrying capacity. Now we've been looking at a growth curve on a logarithmic scale. Turns out bacteria are not the only populations that are influenced by the carrying capacity of their environment. When the carrying capacity is met, the population can only stay at stationary phase for a period of time, and then it will begin to go down. So some of the cells were dying during stationary phase. The primary factors causing them to die is lack of nutrient and an increase in toxic metabolic waste. We're going to be doing some setup for food microbiology. We're going to make some bad apple wine. We're going to make some sauerkraut. And after a period of time of having the organisms in these glass containers, we will see what happens when they go into death phase. And usually they're going into death phase because they produce a lot of metabolic byproducts that kill them. They produce acids, they produce alcohol. These things aren't good for them. So eventually they're making too much nasty end product. Anyway, exponential growth phase, you see the population decline exponentially. So lack of nutrients, increase in metabolic waste, these are the primary factors that are causing the cells to die. Some of them begin dying in stationary, more of them die during the death phase. In death phase, their numbers are really going to decrease rapidly, and oftentimes they will drop below the initial number that you put in there. But if you have bacteria in a container like a flask or a tube, a lot of times they don't all die. Conditions in that container are pretty nasty, but some of the cells actually survive. So here we see kind of a dotted line. This is below the number that was introduced in the first place. And they're down here, they're kind of leveling off. They're just kind of hanging in there. They're not doing real well, but there's some interesting studies that have been done looking at bacteria that are still alive in that batch culture at the end of the exponential death phase. Well, they didn't all die. A limited number can die. And they go into what's called a long-term stationary phase. They've done some studies at UC Davis with salmonella. They can keep them in containers for years in this kind of long-term stationary phase. It's kind of a survival mode that they will go into, and they apparently begin to produce what are called stress response proteins. They're going to make some new enzymes that they weren't making before. They're going to run some new metabolic pathways. Sometimes they become more capable of picking up DNA from their environment. So it's like, oh, those cells died, let's take nutrients from them. So the ones that can survive, they're kind of superbugs. They get to hang out in there and wait for the conditions to improve. But if we were to draw that bacterial population curve and we didn't put it on a log scale, we'd get something that looks like that. Anybody recognize that curve? It's got some dates on it, but this is 1800, 1930, 1960, 1975, 1987, 1999, something like that.
Human population?
That's the human population growth curve, which is a little bit scary because when carrying capacity is reached, the next phase is exponential death. We don't know what the carrying capacity of this planet is, but you can be pretty certain you're not getting off anytime soon. I know there's talk of go to Mars or go someplace else in space or go into a space station. We can't even take care of the planet we're on. You're not going to get off this rock and go someplace else. So this curve has some very important connotations. This is showing human population growth, and we're not the only things on the planet. We are pushing the carrying capacity of this planet, and we are not the only organisms living here. We have to share. Microbes might make us do it. Basically, all populations are limited by carrying capacity. So what does this mean? If you are a young human of reproductive age, please take this seriously. We should not be reproducing like E. coli in a flask. They can't think. They don't know the negative consequences of their actions. We should know. We should be paying attention. But surprisingly, very few humans are. So here's something you can do to educate yourself. It's worth 5 points extra credit. You can go to any one of these websites. The United Nations Population Fund, the Population Connection, the Population Institute. These addresses are in the lecture notes. Go to one of these, go to all three of these, and see what they have to say. What I would like to see happen is anytime any news, whether it's Fox News or MSNBC or whatever, in the corner, instead of showing us what's happening to the stock market, or instead of having that little information thing coming across the bottom, you should have the number ticker that's showing us what's going on with human population. It should be in everybody's face all the time. Because human population is a serious threat to our existence. This Planned Parenthood, it's not a political issue. It's a survival issue. If we don't start planning, we're going to destroy ourselves by overpopulating the planet. And it is already happening. All you have to do is read a little bit and you know it's already happening. Fisheries. People are not catching the same big fish they used to catch. Why? Because the fish populations have been depleted. Other organisms, the orcas are starting to kill off the narwhals. Why? Because the orcas are starving. They can't eat anything else because we've taken all the fish. Then we've caused climate change, which is destroying farmland. And then when the ice melts on the ice caps, we're going to flood California's farmland. I don't think they're going to put a gate across where the Golden Gate is, really. But maybe, because otherwise the valley's going to fill up with salt water. And then it's going to be tough to grow crops. This is a serious, serious issue. We need to stop, like I said, stop reproducing like E. coli in a flask. Tough choices. And it's something that no politician will touch ever. They will not talk about it. Because nobody wants to know. Talk about your inconvenient truth. Oh my word. Too many of us. Anyway, back to bacteria. Bacteria can be grown in what we call continuous cultures. Yeah, on that happy note, let's go here. These are basically set up if you want to grow bacteria and you want to harvest some particular product. So what you do is set up this continuous culture, you're constantly putting food in and you're constantly taking effluent out. And the cells are then maintained in exponential growth phase. You're getting rid of the excess constantly. We don't have any of those in here. But again, it's often used if you're trying to get the organism to produce some kind of product that is necessary or something that you want to harvest. A lot of times it's some enzyme product or something else that they're making. Are you asking a question?
Yeah, so what did you want that extra credit?
Huh?
What did you want specifically for that extra credit?
What? A couple of pages. I want you to show me that you actually went to one of the sites and read some information that they had to say. And you can comment on it in any way that you feel you want to. I don't knock people down for telling me things that I don't want to hear. But I do want you to go to the sites and look at them, because a lot of people, most people that do this at extra credit assignment, what they say is, I had no idea. That's what most people say. Which is kind of news to me, because it's like, really? You haven't noticed the long lines on the freeways? You haven't noticed the crowding? The houses all over Placer County? Scary stuff. Well, anyway. Where are we? So why do we care about population density? Yes.
Was there a due date on that?
Was there a what?
A due date?
Oh, there's never due dates. Well, actually, there is a due date. It's the end of the semester.
Okay.
Everything has to be in. We do reach a point where it's like, okay, stop time. Okay, so why do we want to know about microbial numbers? If you are a dairy farmer and you're bringing milk to a dairy or to a bottling plant, they're going to want to know does your milk have high levels of contamination or low levels of contamination? So they're constantly checking. They're checking the milk and they're looking for probably organisms that might be pathogenic. You want those contaminant levels to be low. Another place where it might come into play, if somebody takes a sample of cerebral spinal fluid, they kind of want to know how many bacteria are in there. There shouldn't be any. So if they're in there, they want to know. They also want to know, are they viable? Are they alive? There was not so long ago an incident where some kind of an additive that was put in peanut butter had salmonella in it. Somebody should have been checking that, because instead they put the additive in and contaminated a whole bunch of peanut butter and then sold it to people, and a lot of people got sick. Well, now we can look at growth on solid media. As I said, a colony forming unit can be one cell, or it could be more than one cell if they have a consistent arrangement. But colonies have a characteristic size and shape, and you might wonder why. Why don't they just keep growing? So here we see colonies on an air plate. They have reached a certain size, and if this air plate was stuck in a drawer, it would continue to look just about like that, probably for as long as the medium was dampish, and then it would kind of dry up. And if you leave them in the incubator, they form what we call auger chips, because all the water evaporates and then the whole thing just dries up. But the colonies tend not to get bigger than that. And the reason is bacteria are dumping enzymes into their environment. They produce a lot of exoenzymes. The exoenzymes digest food material outside the cells, and so they can take apart polysaccharide and then take in the monosaccharide. But what it means is there's not that much nutrient outside the colony. So they really have depleted. Now this colony, it looks like there's lots of agar available, but the nutrient is actually gone. So they've utilized material even outside the colony all the way around. Consequently, they will reach a certain size and stop growing. That's why when we do our colony morphology, we include size. When they're really crowded, they're really little, because they've depleted the nutrient around them. They simply can't grow any bigger. So when you're measuring your colonies, you should measure the largest ones. That's as big as they normally would get. Beyond that, they can't grow because they've taken up the nutrients outside. Now, there are exceptions if they're modal. Then they can do what's called swarming, where they will spread out across the agar surface. Sometimes filamentous swarms do this, but here you can see swarming. I believe these are bacillus colonies. They are swarming this way. Here, see these waves? If you were to just glance at this plate, you might not think there's anything on that. But the cells are swarming. Instead of forming nice discrete colonies, they're just running across the medium. And then we have one morphological unknown that does this. It's impossible to streak a plate and get isolated colonies. They're all just going to swarm across, making these filaments. Now, if you wanted to maintain pure cultures, which you're going to be doing with your semester projects once you get to this point, you should probably transfer them at least every 2 to 3 weeks. It depends, because some cultures will die before that. We try to take care of this by setting up a freezer backup. So we'll have some tubes we keep in the freezer. In case they die, we can go back and get them out. But you have to keep re-streaking them to keep them fresh. The other thing is to recognize, if I back up for a moment, I have to back up more than that. See this colony here, where this sample is taken from the outside edge? When you're taking a sample from a colony, that's where you should take the cells. Take them from the outside edge of the colony. Don't take them from the middle, because the cells in the middle are the oldest. And even though the colonies, they can be completely dead, and they won't look different, but they will lose their capability of growing. And again, that's why we prepare the freezer backups. But you should also always take your sample from the outside edge. That's where the most lively cells will be. And if you re-streak and you get no growth, maybe they died, or maybe they're missing their friends. We run into that sometimes. Well, that's the end. That's good. We're out of time. Okay. No quiz. Woo-hoo.
Yeah, and let's sit at the table and eat and
No,
Let's take this note.
no, just this material. Exactly. So, a fairly significant point, which was just brought up, next Tuesday's quiz covers just this lecture. That's good. So you have less material to study, but please study. I realize some of this is review, but you do have a quiz next Tuesday, even though you have an exam on Thursday.
What about the lab? How many labs is that quiz gonna be on
The lab quiz,
for next week?
Yeah, because
I'll have to think about that, because we have protista and multicellular parasites, and then we're gonna have the food microbiology, but we're not gonna finish the food microbiology. I think next week's quiz should be organisms. So next week's lab quiz, the question is, what should we do with next week's lab quiz? Tonight, we're doing food microbiology, but we're starting food microbiology, we won't finish it. And I think it would be good to have you guys study a little bit and have the lab quiz for next week on multicellular parasites. But we did protista, huh? We can't really just have just multicellular parasites.
Sure we can.
Well, you'll see when we do tonight's presentation on food micro, some of it, even though we don't finish it, there could be some questions from that. So probably a combination of that.